Team number 7, Clostridium Baccilus. Plan of the team, properties of Clostridium bacilli, and epidemiology of tetanus, gas gangrene, botulism, and anthrax, pathogenesis of these diseases, and clinical manifestation of these diseases, and laboratory diagnostic methods. Classification and nomenclature: Clostridium, family Bacillaceae, gene Clostridium, species Clostridium perfringens, Clostridium novi, Clostridium septicum, and Clostridium histolyticum, and Clostridium botulinum, Clostridium tetani. Clostridium botulinum, cause of botulism disease, and Clostridium tetani, cause of tetanus disease. Clostridium perfringens, cause of gas gangrene disease. Clostridium perfringens, gram-positive bacilli, motionless, and spores are oval, terminally situated. Clostridium perfringens is a commensal in large intestine of human beings and animals. It is also found as saprophyte in soil, dust, and air. It is capsulated, non-motile, gram-positive bacillus. It bears subterminal bulging spores, but doesn't produce spores in tissues or in culture media. It is invasive as well as toxigenic. And virulence factors produced by Clostridium perfringens can be grouped into different types: 4 major toxins: alpha, beta, epsilon, and iota. They also produce heat-label enterotoxin and 8 minor toxins: gamma, delta, lambda, kappa, theta, eta, and mu, nu toxins. And the soluble substances are produced such as neuraminidase, histamine, and circulating factor inhibit phagocytosis. And the next, Clostridium. This slide shows types of Clostridium. Laboratory diagnosis of gas gangrene. Specimens are necrotic tissues, muscle fragments, and exudates from a deeper part of wound where the infection appears to be more active. Blood culture may be positive for Clostridium perfringens and Clostridium septicum. Clostridium perfringens bacteremia can occur even the absence of gas gangrene. Swabs rubbed over the wound surface or soaked in exudates are not satisfactory. Specimens should be put into Robertson's cooked meat broth and transported immediately to the laboratory. Direct microscopy, gram-stained films provide clues about the species of Clostridia present. Absence of neutrophils in the infected tissues is a characteristic feature. And the thick, stubby, boxcar-shaped gram-positive bacilli without spores suggestive of Clostridium perfringens. And the spore-bearing gram-positive bacilli suggest other Clostridia species. And gas gangrene, clinical manifestation of gas gangrene. The incubation period is variable depending upon the nature of injury, the amount of wound contamination, and the type of Clostridial species involved. The incubation period varies. For example, 10-48 hours for Clostridium perfringens, and the various manifestations include sudden onset of excruciating pain at the affected site, rapid development of foul smellings in serosanguineous discharge and gas bubbles, crepitus in the muscle plants, and browny edema and induration. Such gangrenous tissues later may become liquefied and sloughed off, and shock and organ failure develop later, associated with higher mortality rate, 50%. Laboratory diagnosis: specimens, necrotic tissue, muscle fragments, and exudates from deeper part of the wound. Culture method: Robertson cooked meat broth, egg yolk agar, and incubation anaerobically by gas pack. And tetanus disease, Clostridium tetani, cause of tetanus disease. Clostridium tetani is an obligate anaerobic gram-positive bacillus with terminal round spore drumstick appearance. It is the causative agent of tetanus, an acute disease manifested by skeletal muscle spasm and autonomic nervous system disturbance. Tetanus has been known since an ancient time, and virulence factors of Clostridium tetani produces two exotoxins, tetanolysin and tetanospasmin. Tetanolysin, it is a heat-label oxygen label hemolysin, antigenically and related to the oxygen label hemolysins produced by Clostridium perfringens and Streptococcus pyogenes and Streptococcus pneumonia. It plays no role in the pathogenesis of tetanus. Tetanospasmin or tetanus toxin, it is a neurotoxin responsible for disease manifestation. It is oxygen stable but heat label. Toxin is produced as a single 150 kilodalton polypeptide chain that is cleaved to produce heavy and light chains joined by a disulfate bond. And it is antigenic and is especially neutralized by its antitoxin. It gets toxoided spontaneously or by formaldehyde. The toxoid form is antigenic but loses virulence property, hence it is used for vaccine preparation. And tetanus toxin binds to receptors and present on motor nerve terminals, which results in toxin internalization. And the following internalization, tetanus toxin gets transported in retrograde way to the gamma aminobutyric acid and glycine producing inhibitory neuron terminals, and the toxin prevents the presympathetic release of inhibitory neurotransmitters glycine and GABA, which leads to spastic muscle contraction. Epidemiology of tetanus. Mode of transmission: tetanus bacilli enter through injury, superficial abrasions and punctured wounds, road traffic accidents, surgery done without proper asepsis, and neonates following abortion due to unhygienic practices and otitis media, and it is non-infectious. There is no person-to-person spread. Clinical manifestation: incubation period is about 10 days, and the shorter the incubation period, graver is the prognosis. Muscles of the face and jaw are often affected first, and first symptom increase in the masseter tone leading to trismus or lockjaw, and followed by muscle pain and stiffness, back pain, and difficulty in swallowing. In neonates, difficulty in feeding is the usual presentation. As the disease progresses, painful muscle spasm develops, which may be localized, involves the affected limb, and generalized painful muscle spasm leads to descending spastic paralysis, and hence feet are spread and mentation is unimpaired. Deep tendon reflexes are are exaggerated, and next is autonomic disturbance is maximal during the second week of severe tetanus, characterized by low or high blood pressure, tachycardia, intestinal stasis, sweating, increased tracheal secretions, and acute renal failure. Tetanus pathogenesis of this disease. This is the penetration of the pathogen through the entrance gate various wound, and the next process is pathogen produce toxin through the motor fibers of the peripheral nerves, and through the blood enters the spinal, and the medulla oblongata is fixed in the synapses, and the next process continuous supply of exciting pulses to the muscles, and it leads to tonic muscle tension. As pathogenesis of tetanus is toxin-mediated, the association of the isolated organism can only be established when its toxin production is demonstrated. Toxigenicity can be detected by both in vitro and in vivo methods. In vitro hemolysis inhibition test, Clostridium tetani produces hemolysis on blood agar, which is inhibited by adding antitoxin. This test indicates the production of only tetanolysin but not tetanospasmin. In vivo mouse inoculation test, and Robertson's cooked meat broth with black turbid growth is injected into the root of the tail of test mouse. The test animal develops stiffness, which begins with the tail and progresses to involve the hind limbs on the inoculated side, leads to the other limb and the next trunk and the next four limbs. Death occurs within 2 days. This test indicates the production of tetanospasmin. This slide shows shape of Clostridium tetani. Culture is more reliable than microscopy. Robertson cooked meat broth, Clostridium tetani being proteolytic, turns the meat particles black and produces foul odor. And the blood agar with polymyxin B. These plates are incubated at 37 degrees Celsius for 48 hours under anaerobic condition. Clostridium tetani produces characteristics swarming growth. And the tetanus prevention, diphtheria and tetanus vaccines we use, and diphtheria, tetanus, and pertussis vaccines we use, tetanus and diphtheria vaccines, and tetanus toxoid is commonly used for active immunization. It is available either as monovalent vaccine, plain formal toxoid. Toxoid is prepared by incubating toxin with formalin, and the primary immunization of children. Tetanus toxoid is given under National Immunization Schedule of India, and total 7 doses are given. 3 doses of DPT. This diphtheria, tetanus, and pertussis at 6, 10, and 14 weeks of birth, followed by 2 booster doses of diphtheria, pertussis, tetanus at 16, 40, and 16-24 weeks and 5 years, followed by 2 additional doses of TT at 10 years and 16 years. Adult immunization is primary immunization is not administered in childhood, then adults can be immunized with tetanus toxoid. 4 doses of TT is given 2 doses of TT and at 1 month interval, followed by 2 booster doses at 1 year and 6 years. TT is tetanus toxoid. Characterization of anaerobiosis. Anaerobes don't use oxygen, they don't have cytochromes and cytochrome oxidase. Anaerobes don't decompose hydrogen peroxide due to the lack of catalase and peroxidase. Anaerobes may accumulate hydrogen peroxide because the enzyme peroxide dismutase is absent. Etiology of botulism. Next disease about botulism, Clostridium botulinum. Clostridium botulinum produces botulinum toxin and causes botulism, rare disease manifested as various clinical syndromes ranging from food poisoning and wound infection to infant botulism. Clostridium botulism is anaerobic gram-positive bacillus with subterminal spore, and it is ubiquitous in nature, widely distributed as saprophyte in soil, animal manure, and vegetables and sea mud. And pathogen Clostridium botulinum, vegetative and spore forms. There are 7 types of pathogen, serotypes and A, B, C, D, E, F, G, and vegetative form anaerobes. They produce exotoxin, neurotoxin, hemagglutinin, and protectin. And pathogenesis of botulism disease. Clostridium botulinum is non-invasive. Its pathogenesis is due to production of powerful neurotoxin, botulinum toxin, probably the most toxic substance known to be lethal to mankind. Botulinum toxin is a 150 kilodalton dependent protein consisting of 100 kilodalton heavy chain and 50 kilodalton a light chain. Serotype: the light chain is serotype specific and can be typed into 8 serotypes: A, B, C1, C2, D, E, F, and G. Serotypes A, B, E commonly cause human disease, most severe being serotype A, and all serotypes produce neurotoxin. Botulinum toxin types C and D are bacteriophage coded. Botulinum toxin differs from other exotoxins as it is produced intracellularly, not secreted, and appears outside only after autolysis of bacterial cell. Toxin is synthesized initially as a non-toxic protoxin. It requires trypsin or other proteolytic enzymes to convert it into active form. Mechanism of action of botulinum toxin: it produces flaccid paralysis. Mechanism of action of botulinum toxin: after entry, botulinum toxin is transported via blood to peripheral cholinergic nerve terminals. The most common nerve terminal sites are neuromuscular junctions and the postganglionic parasympathetic nerve endings and the peripheral ganglia. It doesn't affect the CNS, central nervous system. Botulinum toxin binds to acetylcholine receptors on the nerve terminals at neuromuscular junction, which results in blockage of release of the acetylcholine, leading to flaccid paralysis. Clinical manifestation of this disease, the manifestation of botulism, are due to decreased acetylcholine in cranial nerve and the parasympathetic nerve terminals. Common symptoms include diplopia, dysphagia, dysarthria, and descending symmetric flaccid paralysis of voluntary muscles and decreased deep tendon reflexes, and there is no sensory or uh, and cognitive deficits, and respiratory muscle paralysis may lead to death, and constipations. And the types of botulism: foodborne botulism, wound botulism, infant botulism, and adult intestinal botulism, and the iatrogenic botulism. Gene Baccilus, species Baccilus anthracus. Morphology of Baccilus anthracus: bacilli 5 micrometer and edges chopped off in strokes in pairs or chain, non-motile. The capsule is formed in the host organism, oval spore, central location, and gram positive. And anthrax, a smear from an old pearl culture of anthrax bacilli. According to Zil-Nielsen, stained blue sticks, red spores. Spore location is central, and the cultural properties. It is an aerobe and facultative anaerobe with a temperature range for growth from 12 to 45 degrees Celsius, optimum 37 degrees Celsius, and the good growth occurs on ordinary media. On agar plates, and irregularly round colonies are formed from 2 to 3 millimeters millimeters and in diameter, raised dull opaque and grayish white with frosted glass and appearance. And under the low power microscope, the edge of the colony is composed of long interlacing chains of bacilli resembling locks of matted hair. This is called the medusa head appearance. Capsule culture specimen collections. The specimens should be collected before starting antibiotic treatment. The useful specimens are pus or swab from malignant pustule and sputum in pulmonary anthrax, blood in septicemia, spinal and cerebrospinal fluid in hemorrhage, meningitis, and gastric aspirate, feces or food in intestinal tracts, and ear lobes from dead animals. And the direct demonstration gram staining method reveals gram positive large rods. Spores are usually not seen in clinical samples, and polypeptide capsule can be demonstrated by staining with Gurs polysacchrom, polychrome methylene blue stain for 30 seconds, and capsule appears as amorphous purple material surrounding blue bacilli. This is used for presumptive diagnosis of animal anthrax. And the direct immunofluorescence test. It detects capsular and cell wall polypeptide antigens by using fluorescent target monoclonal antibodies. It is used for confirmation of the diagnosis during bioterrorism outbreaks. Biochemical properties of this pathogen. Biochemical properties is specimens pus, sputum, blood, CSF we take, and we need a blood agar and nutrient agar, and next is we need gelatin stap agar. Growth occurs as inverted fir tree appearance due to liquefaction of gelatin, which occurs maximum at the surface and then slows down towards the bottom. And spore staining and lipid granule staining with Sudan black B, and gram staining, gram positive, large bacilli, rectangular bacilli, and we use direct immunofluorescent method, and we use serology, molecular diagnosis, PCR. Polymerase chain reaction with specific primers can be used for further confirmation. And next is Baccilus anthracis ferments glucose, sucrose, maltose, and dextrin with the production of acid, but no gas. Nitrate is reduced to nitrate, and catalase is produced, and the Vogus-Proskauer test is positive. Pathogenicity factors: capsular polypeptide, the capsule which is polypeptide of D-glutamic acid inhibits phagocytosis, and anthrax toxin, it is a plasmine-coated toxin complex comprising of 3 protein components: protective antigen, lethal factor, or edema factor. And the cutaneous form of anthrax on the skin, and this anthrax on the skin, black-case chair is surrounded by a ring of vesicles containing serous fluid and an area of edema, and in duration which may become extensive. And the pulmonary form of anthrax, spores in alveolus, and the damage, Bacillus anthrax damage of lung tissues. And pulmonary type of anthrax, wool sorters disease, as it is seen in workers of wool factory, acquire infection by inhalation of dust from infected wool and inhalation of spores. Hemorrhagic pneumonia develops, and bacilli spread by lymphatics or blood, leading to bacteremia, hemorrhagic mediastinitis, and hemorrhagic meningitis. Workers of occupational exposures, workers of wool factory, and prognosis fatal, and bioterrorism, most common form of cause bioterrorism. And the intestinal form of anthrax. It is rare, occurs due to ingestion of spores contaminated with meat or animals dying of anthrax, and it is highly fatal and manifested as bloody diarrhea. Agent Baccilus anthracis is one of the most common agents of bioterrorism. It has been widely used in various biological warfare, such as outbreaks and next, and laboratory diagnosis of anthrax. Laboratory diagnosis of anthrax. We need specimen pus, sputum, blood, and cerebrospinal fluid, and direct demonstration gram staining method, gram positive large rectangular bacilli, and McFadden's reaction shows amorphous purple capsule surrounding blue bacilli, and direct immunofluorescence and detect capsular antigen, and the Ascolis thermoprecipitation test we use, and culture smear gram staining reveals gram positive rods with bamboo stick appearance, spore staining, Ship's method, and acid fast staining, and lipid granular staining with Sudan black B, and antibodies detection by ELISA, enzyme-linked immunosorbent assay, and molecular diagnosis PCR, detecting specific genes of the Baccilus anthracis, and molecular typing we use. Molecular typing, it is useful for epidemiological studies to trace the source of infection. Various methods of available are as follows: multiple locus, variable number of tandem, repeat analysis, and amplified fragment lengths polymorphisms.